Imagine you feel, I don't know, a headache coming on, so you head to the medicine cabinet and you just swallow an ibuprofen.
Right.
Or you're just waking up, you're pouring that first cup of morning coffee and taking a sip. We treat these actions as entirely mundane.
Oh, complete.
But beneath the surface, you are, introducing a foreign chemical into this chaotic, acidic water slide of your digestive system. And somehow, reliably, that chemical navigates the internal labyrinth, finds its target, and stops your head from throbbing or, clears your mental fog.
It's really a localized biological intervention,
Huh.
but it's operating on a massive systemic scale. And honestly,
Uh,
for most of human history, that process felt indistinguishable from magic. I mean, we observed the outcomes without having any concept of the underlying machinery.
Okay, well, let's unpack this machinery because that is the mission of today's deep dive. We are looking at a massive stack of pharmacology research to understand exactly what drugs do to the human body. This is the field of pharmacodynamics.
Yeah. And to set the stage for you, we really need to draw a hard line between two terms you will constantly see in the literature, pharmacokinetics, or PK, and pharmacodynamics, or PD.
Right. Okay, so a standard memory trick here. Let's unpack this. Pharmacokinetics is what the body does to the drug.
Exactly. It covers absorption, liver metabolism, excretion, all of that.
But pharmacodynamics, which is our focus today, is the exact inverse. It's what the drug does to the body.
Yes. At a fundamental level, we are talking about receptor binding and dose-response relationships. It's basically the math of how a specific concentration of a drug at a specific cellular site translates into a physical effect.
Though looking at the sources, modern pharmacology has pushed that definition much further, hasn't it?
Oh, absolutely. We're now operating in the realm of systems pharmacology. So we don't just look at a chemical hitting a single receptor in a vacuum anymore. We have to model how that interaction ripples across entire cellular networks.
Huh.
In fact, regulatory agencies, like the FDA, they actually consider long-term adaptive changes to be central to a drug's pharmacodynamics.
So wait, we're not just looking at the immediate relief of the headache then. We're looking at, I guess, what happens to the cellular architecture if you take that drug every day for a year.
Precisely. Take the concept of downregulation, for example.
Mm.
When you constantly bombard a cell's receptors with a high concentration of a drug, the cell doesn't just passively accept the overstimulation.
It fights back.
Basically, yeah, it physically adapts. So through a process called endocytosis, the cell will literally swallow its own receptors.
Wait, swallow them? Really?
Yeah, it pulls them beneath the cell membrane so the drug can no longer access them. And this is the mechanical basis of building a tolerance to a medication.
Oh, wow.
That structural remodeling, it isn't just some side effect of pharmacodynamics, it's a core feature that determines if a treatment actually remains viable long-term.
That is wild. But to understand how we even mapped out these cellular mechanisms in the first place, we have to track the evolution of the science itself. Because up until the mid-19th century, medicine was almost entirely descriptive.
Right, it was just cataloging clinical anecdotes, like noticing that chewing a specific bark reduced a fever, but having absolutely no idea why.
The transition to mechanistic science required a radical shift, which started with Rudolf Buchheim. He founded the first Pharmacology Institute in 1847.
Yeah, he argued that just cataloging folk remedies was, well, insufficient. Researchers had to actually induce drug effects experimentally.
And his student, Oswald Schmiedeberg, kind of established the template we still use today, right?
Yeah,
Isolating a specific chemical action and tracing it to a specific biological tissue,
exactly.
which perfectly sets up that famous 1850s experiment by the French physiologist Claude Bernard. He was studying curare, the plant-based paralytic they used on blow darts.
Right, a classic experiment.
The medical community knew curare paralyzed the body, but the actual site of action was a total mystery. So Bernard takes a frog's leg, isolates it, ties it off to stop the blood flow, but he leaves the nerve intact and then applies the curare.
It was just a brilliant exercise in systematic elimination.
I always think of it like a broken lamp in your living room. You don't just assume the entire house is broken. You check the light bulb, then the power cord, then the wall outlet. You test each component to find the specific point of failure.
That's a perfect analogy. And Bernard essentially tested the biological components. He stimulated the nerve above the tie-off no muscle movement.
Right.
Then he stimulated the muscle directly, and the muscle twitched.
Proving that the nerve fibers were functional and the muscle tissue was functional.
Yes. So the paralysis was isolated entirely to the junction between the nerve and the muscle.
A cord worked, the bulb worked, but the connection point was blocked.
Exactly. and it was the first time anyone had localized a drug's pharmacodynamic action to a specific cellular junction.
But even though Bernard localized the site, it took several more decades to develop like a molecular vocabulary for what that junction actually was.
Right. That brings us to 1878. Cambridge physiologist John Newport Langley was experimenting on cats using two plant derivatives, atropine and pilocarpine.
And he noticed they had entirely antagonistic effects on the cat's salivary glands, right?
Yeah. One stimulated saliva production, and the other completely suppressed it. Crucially, though, introducing one could physically displace the biological effect of the other.
Meaning these two totally different molecules were competing for the exact same biological parking spot.
They were fighting for a shared finite structure in the tissue. Langley eventually called this hypothetical site a receptive substance.
Receptive substance, right?
And shortly after that, the immunologist Paul Ehrlich codified this concept with his side chain theory. He famously stated in Latin that agents do not act unless they are bound.
The drug molecule is the key, and the body's receptor is the lock.
Exactly. That lock and key visualization drove Ehrlich to hunt for what he called a magic bullet. Basically, a chemical engineered to bind selectively to a disease organism while ignoring healthy host tissue.
Which led to his 1909 discovery of salvarsan.
Right, an arsenic-based compound that targeted syphilis without killing the patient. What's fascinating here is that this exact template, identifying a receptor and synthesizing a molecule to fit it, remains the foundational template of all pharmaceutical research today.
The receptor is the physical basis of pharmacological specificity.
Yes.
So once we establish that drugs must physically bind to receptors to exert an effect, the math of pharmacology really had to catch up. Like, how do we actually quantify the relationship between the number of molecules you ingest and the scale of the biological response?
Well, in 1926, the pharmacologist A.J. Clark formalized this with occupancy theory. He leaned heavily on the law of mass action from chemistry. Clark proposed that drug-receptor interactions function as a simple chemical equilibrium. So the higher the concentration of the drug, the more receptors are occupied, and the greater the biological effect. This gives us that classic S-shaped dose-response curve.
And two distinct metrics define that curve. This is actually where we need to clear up a major public misconception for you listening. When people describe a medication as strong, they usually conflate two completely separate variables, right? Potency and efficacy.
Oh, constantly. They treat them as interchangeable, but on a pharmacological graph, they represent different axes entirely.
Let's use a car analogy. Potency, which is measured as the EC50 on that S-curve, is basically how lightly you have to tap the gas pedal to get the engine revving.
Right.
A highly potent drug requires only a tiny microgram to initiate a biological response. But efficacy, or Emax, is the car's absolute top speed. It's the maximum physiological effect the drug is capable of producing, regardless of how much you fled the engine.
That's spot on. Let's apply that to clinical pain management. Aspirin and morphine are both analgesics, but morphine possesses a dramatically higher efficacy ceiling.
Right, they aren't even in the same league.
Exactly. If a patient is recovering from major abdominal surgery, no amount of aspirin is going to manage that pain. A physician could increase the aspirin dose to its absolute maximum, saturating every single available receptor to maximize its potency. But the drug simply lacks the efficacy to suppress severe neural pain signals the way morphine does.
So if efficacy represents the maximum effect a drug can trigger once it parks in the receptor, we really need to look at the mechanics of what happens after the binding occurs. Because plugging the key into the lock doesn't always open the door in the exact same way.
No, it doesn't. We actually classify these interactions across the spectrum of action. At the very top, we have full agonists.
Okay.
These molecules bind to the receptor and induce a shape change that activates the intracellular signaling pathway to its maximum possible degree. Fentanyl acting on the mu-opioid receptor is a classic full agonist.
And then we have partial agonists.
Right. These bind to the exact same receptor, but their physical structure doesn't perfectly align the receptors to internal proteins. So even at 100% receptor occupancy, they can only trigger a fraction of the maximum biological response.
So the natural consequence of that is you get like a built-in safety ceiling.
Exactly. Buprenorphine, which is used in opioid use disorder treatment, is a classic partial agonist. Because it cannot fully activate the opioid receptor, it severely limits respiratory depression.
Oh, that makes sense.
Yeah, it prevents withdrawal symptoms, but it won't suppress the patient's breathing past a certain threshold, which mitigates the primary cause of fatal overdoses.
That's brilliant. And then we move to competitive antagonists. These molecules bind to the receptor, but they lack the structural features necessary to trigger any intracellular response. They just occupy the space. And here's where it gets really interesting, because this challenges the notion that antagonists don't do anything. Take naloxone or Narcan. If a patient is experiencing a fentanyl overdose, the fentanyl is acting as a full agonist, shutting down the respiratory drive. Then you administer naloxone, which is a competitive antagonist.
And naloxone possesses a significantly higher binding affinity for the mu-opioid receptor than fentanyl does.
It's a much stronger chemical magnet, so it rapidly displaces the fentanyl, literally kicking it out of the receptors. And since it doesn't activate the receptor itself, the respiratory suppression stops, and the patient wakes up within minutes.
It's incredible to witness.
Claiming an antagonist doesn't do anything is honestly like claiming a bouncer at a nightclub door isn't doing anything. I mean, sure, the bouncer isn't inside dancing, but by physically occupying the doorway and aggressively blocking anyone else from entering, they dictate the entire environment of the club.
That's a really good way to put it. Blocking is a highly active therapeutic effect. But to fully appreciate receptor mechanics, we have to look at inverse agonists, which are just mind-bending.
Well, because an antagonist just drops the drug-induced activity back to a neutral zero, right?
Correct. But an inverse agonist suppresses receptor activity below its resting baseline.
Which implies that the receptor is biologically active even when the room is entirely empty.
Exactly. That was a massive paradigm-shifting realization in the field. Many cellular receptors exhibit spontaneous constitutive activity.
Like a hum.
Yeah, to continuously hum, firing off low-level signals even in the total absence of any endogenous chemicals or synthetic drugs. An inverse agonist binds to the receptor and forces it into an inactive shape, actively silencing that baseline hum.
That structural reality completely changes how we view cellular communication. But let's bring this theoretical receptor math into the clinical environment, because plotting an elegant S-curve in a petri dish is one thing, but introducing that chemical into a genetically diverse human population is another.
It's much messier.
What happens when the dose required to trigger a therapeutic effect is dangerously close to the dose that triggers systemic toxicity?
Well, we evaluate that risk using the therapeutic index. It quantifies the margin of safety between the effective dose and the toxic dose. Penicillin, for example, has a massive therapeutic index.
Right, you can take a ton of it.
Yeah, a physician can administer a dose significantly higher than the standard requirement with negligible risk of toxicity. But then you have digoxin, a heart failure medication derived from the foxglove plant, and it operates on a terrifyingly narrow index.
Why is digoxin so restricted?
Because the binding affinity of digoxin for its target enzyme in the heart muscle overlaps heavily with its affinity for secondary pathways that cause fatal arrhythmias. The dose that strengthens cardiac contractions and the dose that induces a toxic cascade are separated by roughly a factor of just 2.
Wow, just a factor of 2.
Yeah, minor fluctuations in a patient's blood concentration can easily push them from therapy straight into toxicology.
If we connect this to the bigger picture, it really recontextualizes the Renaissance physician Paracelsus's famous rule: the dose makes the poison.
Exactly.
That isn't just some philosophical observation, it is a rigid pharmacodynamic rule. Toxicology is basically just pharmacodynamics operating beyond the therapeutic window.
And the scary thing is, that therapeutic window is not fixed. It shifts dramatically based on individual genetics. It's an entire area of study called pharmacodynamics.
The blood thinner warfarin is a perfect example of this for you. The goal of warfarin is to inhibit the VKORC1 enzyme, which plays a key role in blood clotting. But the specific genetic code that builds your VKORC1 enzyme varies across the human population.
Right.
If your genetic variant produces an enzyme with a slightly different physical shape, the warfarin molecule cannot bind as effectively.
The lock basically has a slightly different internal configuration.
Right, so the standard dose-response math completely falls apart. One patient might require 10 times the baseline dose just to achieve sufficient receptor occupancy to prevent a stroke. But if you give that massive dose to a patient with the standard genetic variant, the intense receptor blockade will cause severe internal hemorrhaging.
Your DNA continuously redraws your personal dose-response curve.
It's wild.
And honestly, this biological variability is exactly why theoretical receptor modeling has occasionally led to absolute catastrophic clinical failures. For decades, pharmacologists relied on A.J. Clark's occupancy theory, the assumption of a linear relationship between occupied receptors and biological effect. But biological systems just don't behave like simple math equations.
But wait, if mass action dictates that more drug equals more effect, how can that linear relationship actually be wrong?
Because of intracellular amplification cascades, researchers discovered this phenomenon called spare receptors.
Spare receptors.
Yeah. In many tissues, activating a single receptor might wake up a G protein, which then activates 100 secondary enzymes, which produce 10,000 signaling molecules. Because the internal amplifier is so incredibly powerful, you can achieve 100% of the tissue's maximum biological response while only occupying a tiny fraction, sometimes less than 10% of the available receptors on the cell surface.
Oh, wow, which means the tissue is vastly more sensitive than a simple one-to-one receptor model suggests.
Exactly. And underestimating that sensitivity is what led to the tragic 2006 TGN-1912 trial in London. Researchers developed a monoclonal antibody, basically a highly engineered homing missile, designed as a superagonist for the CD28 receptor on human T cells.
And the goal was to stimulate the immune system, right?
Right. So they established a safety baseline in animal models and then transitioned to 6 healthy human volunteers. They administered a dose that was 500 times lower than the highest safe dose observed in animals.
Wait, 500 times lower? But if you reduce a dose by a factor of 500, shouldn't the sheer lack of physical molecules prevent a systemic reaction, regardless of how the drug acts? I mean, that's barely anything.
That was the assumption.
Hmm.
But TGN-112 was a superagonist. In animal models, the CD28 receptor required a secondary chemical signal to fully activate the immune cell. But human CD28 receptors feature a microscopic structural variance.
Oh, no.
When the antibody bound to the human receptor, it locked into a permanent on state without needing that secondary signal at all. Even at trace amounts, the amplification cascade was just devastating.
It triggered a cytokine storm.
A catastrophic positive feedback loop, yeah.
Hmm.
The artificially locked receptors cause the T cells to release massive amounts of inflammatory proteins called cytokines. These cytokines recruited more immune cells, which released more cytokines, systematically destroying the volunteers' vascular linings and triggering multiple organ failure within 90 minutes.
God, in 90 minutes. It's a harrowing reminder that a perfectly conserved biological target in an animal model does not guarantee identical pharmacodynamic behavior in humans.
Not at all.
And sometimes, the structural failure isn't even about species differences. Sometimes it's about the drug hitting an entirely unintended target within your own body.
Right, off-target effects.
Which is exactly what happened in the 1990s with the fen-phen scandal. Fenfluramine was prescribed globally as an appetite suppressant. It functioned by interacting with serotonin pathways in the brain. But pharmacodynamics is ultimately just about physical shapes colliding in the dark.
Very true.
It was eventually discovered that the fenfluramine molecule structurally mimicked the key for the 5-HT2B serotonin receptor, which is prominently located on human heart valves.
So the drug successfully triggered the intended brain receptors, but it simultaneously locked into the cardiac receptors, stimulating inappropriate cellular growth that led to severe valvular heart disease.
These historical inflection points are so critical for dismantling widespread medical misconceptions. Let's bust a couple of these right now. The first one being the assumption that natural or plant-derived compounds are somehow inherently gentler or safer than synthetic pharmaceuticals.
Oh, yeah. The mechanics of receptor binding render that distinction completely meaningless. I mean, consider digoxin from the foxglove plant, or atropine derived from belladonna, the deadly nightshade.
Right, completely natural.
Entirely natural, unengineered molecules, yet they operate on human receptors with razor-thin safety margins capable of inducing lethal toxicity. A cell's receptor does not evaluate a molecule's origin. It doesn't care whether it was synthesized in a steel vat or harvested from a forest. It responds exclusively to binding affinity and structural efficacy.
A molecule is simply a structural geometry. And the second major misconception we really need to address is the idea that placebos are purely psychological phenomena, that they exist, in the mind without any real pharmacodynamic mechanism.
This represents one of the most significant paradigm shifts in pain management. Historically, the medical community dismissed the placebo effect as just reporting bias. Basically, patients simply claiming they felt better to please the physician.
Right.
But a landmark 1978 dental study totally dismantled that assumption. Researchers administered a placebo to patients experiencing severe post-surgical pain, and a significant portion reported genuine relief.
At which point, the researchers secretly administered naloxone.
Yes, the opioid bouncer.
They introduced the competitive antagonist.
And suddenly, the placebo-induced pain relief vanished. The patients rapidly returned to their baseline pain states.
Because the naloxone physically blocked the opioid receptors. Meaning the placebo effect wasn't just some psychological trick. The patient's brains had actively manufactured and released real endogenous opioids in response to the expectation of receiving medicine.
Exactly. The placebo response is a highly active, measurable pharmacodynamic event. The biological cascade is literally identical to administering morphine. The trigger is just the cognitive expectation rather than an external chemical.
So, what does this all mean for you listening? When you navigate the medical system, you're constantly navigating these pharmacodynamic principles. When a physician asks if you consume grapefruit juice before prescribing a statin, they're managing a crucial interaction.
Yeah, because grapefruit juice inhibits a specific liver enzyme responsible for clearing drugs from the bloodstream.
Right.
By slowing that clearance, the juice artificially spikes the concentration of the statin bombarding your cellular receptors, which pushes the dose-response curve from a therapeutic level into potential toxicity.
It all comes back to managing that specific concentration at the specific receptor site.
And honestly, this raises an important question and highlights the ultimate reality of pharmacology. You cannot separate pharmacodynamics from pharmacokinetics. A pharmaceutical company can engineer a drug with flawless PD-perfect binding affinity, highly selective target architecture, optimal efficacy. But as the PK is flawed, the drug is useless.
It never reaches the tissue.
Exactly. If stomach acid degrades the molecule before absorption or the liver metabolizes it too rapidly for it to reach the target tissue, all of that elegant receptor engineering is just wasted. The chemical must actually survive the journey to unlock the door.
It is a miraculous, delicate, and deeply complex biological dance happening inside you every single time you swallow a pill. Which leaves us with a final, slightly provocative thought regarding that 1978 dental study. If a purely psychological expectation can compel the brain to manufacture chemicals that physically bind to cellular receptors and alter biological reality, how much of the efficacy of any medication we take is the external compound physically turning the lock, and how much is simply our brain's powerful expectation doing the heavy lifting? The next time you wake up and take that sip of morning coffee, think about those billions of receptors waiting in the dark, responding not just to the chemicals you consume, but to the reality you expect.